Hello, and I'm really happy to be here, and I feel very honored by the fact that I was invited to speak here. In particular, as already Guilhem mentioned, I'm one of the ones who never had the possibility to directly interact with Giorgio, but I, in a sense, remained all my life at Giorgio number 2 or so. But basically, most of my scientific life is at this distance, so there is a lot of influence from Giorgio, and I'm extremely grateful to this, and so I find very, very nice to be able to present my work here. Even more, I want to, a little anecdote. I grew up in Eastern Berlin behind the wall, and in 1989 the wall came down, and I was a young undergraduate student. And at this point, as one little side effect of this wall coming down, I could go just to the other side of the wall, to Western Berlin, to the library. And in the year after, I read or tried to read 2 books. In particular, one was very new. It was called Statistical Field Theory. Came out, I think, the year before. And there was the famous Mesa Parisi Virasoro. I fought quite a bit, but one of the results of the wall coming down is that I'm now here, even married to a Roman, and it's Giorgio's fault, partially at least. Ricardo is not here, he cannot complain that I completely didn't talk about it. Okay, so let me come to my talk. I have picked a citation of George Box, a statistician, as a title. All models are wrong, but some are useful. In particular, my models will be wrong. And it's something George Box told about modeling data, and this is actually really the context I'm going to discuss here. But in a sense, you can use it also as some kind of paradigmatic phrase for all the field of the statistical physics of disordered systems. Because, for instance, as David mentioned, we have spent a lot of time of working on models which we know they are not real models, but they have led us to a lot of insight, and so it's even for statistical physics more in general. But I want to talk about modeling protein sequences, so it will be very applied, much more applied than all the talks we have seen this morning so far. So the starting point is that biological sequence data are accumulating at an extremely impressive pace. You see here a plot of the growth of the so-called Swiss-Prot Database, which is a database which contains all known protein sequences. So if there's a sequencing project, you put the sequences into this database. And you see there is an enormous increase. Actually, you may ask about this kink here, wonder about this kink. And what happened in 2014, more or less, that people realized that this database was coming close to 100 million entries and was kind of hard to keep it sustainable. So they tried to keep it sustainable. And they removed a lot of redundancy. For instance, before there were some bacteria, for some bacteria there were different strains sequenced, so they basically had the same entry several times, so they reduced all this redundancy, and they gained about a factor 2. And then it started to grow again. I just looked up, it's now at 125 million, so we are well behind there. So these are sequences. But there is one sub-database of the Swiss-Prot database, which is the so-called Swiss-Prot database, which is this blue line, which is almost constant at 500,000. And these are all those proteins where we have some knowledge, so some human-made knowledge, so some researcher has worked on this, has found out some information about these proteins which was worth to put into this database. This can be some functional information, this can be some structural information, this can be some mutagenesis studies and so on, can be very different information. And you see, this one stays flat. It does not stay flat because people don't work anymore in biology and don't find out new things about proteins, but basically biologists tend to work on what is called model organisms, coming from Escherichia coli to human, and work on proteins from these model organisms. So they find a lot out about proteins which are already in here, and the annotations which are there, the informations are updated permanently and get better and better. But anyway, the gap between what is known in terms of just the sequence, 20 letters amino acids, and what is known in terms of function, structure, and so on, this gap is opening wider and wider. But the situation is not so bad, and is actually very nice for people who like statistical physics, because actually these data is not completely unstructured data. So you can classify it into what is called protein families. So a protein family, in principle, is defined by a common evolutionary origin. So it's something which is called homology for proteins. So they have there is some common origin, some ancestral protein which is common to all of them. And what is important is that these proteins typically share a lot of their biological function, and in particular also their three-dimensional fold of the protein. So they have very, very similar structure. So you may imagine them, for instance, as basically the same protein doing the same job, for instance, in human and in yeast, or even in E. coli in some cases. But since we have a long time of evolution which happened, they can have very, very different sequences. So the realization in terms of sequence of this structure, of this function, can be very different. So typically we have something like 20 to 30% of the amino acids are in common, and all the others have been exchanged, have been mutated in the course of evolution. Completely random variability would be something like having 5% in common, because we have 20 amino acids, and by chance we would have just 5% in common. So it's more than random, but still it's very variable. And something which is important, these databases do a good job for us in providing us something which is called a multiple sequence alignment. And it's something which looks like this one here, where each line, like the blue one here, is one specific proteins. And they are aligned to be such that a column is a specific protein position, a specific amino acid or residue in all these proteins. And typical sizes of these multiple sequence alignments are something like 50 to 500 positions. So the length of the lines 50 to 500. And we have something like 1,000 to 100,000 lines, depending on which protein family we take. So in PFAM, there are in total 16,000 protein families at the moment, which are more or less well constructed. I think I need to make some little remark on relation to statistical physics, or on some differences to the standard or classical statistical physics of disordered systems. One is something which one is that actually we have this as an input data, so we have different realizations of the same system. That's why it's kind of what we call an inverse problem, because we don't know the model behind there. So we want to construct a model which is actually describing the variability of these sequences, so which could be have generated these sequences in the first place. And we don't even know how this model looks like. And that's why each model we will write down will be wrong, making reference to the title of the talk. And the second point is that in terms of disorder, the disorder in this here is actually what contains the information. Guilhem mentioned this in his talk in inference problems. We have structured disorder frequently, and this structure is actually what we are after. And you see, for instance, in this alignment here, that there is structure. For instance, if you look to this first yellow column, it's made only of H. It's a histidine, it's a specific amino acid which is totally conserved, and actually all these proteins, the family is called histidine kinase because of this H there. And there's another column which wildly changes the amino acid which is present there. So you see that the disorder is actually somehow structured. What I would like to do is to cast these matrices, these multiple sequence alignments, into statistical models. So to something which gives a probability to a full-length sequence, so to one of these lines in the alignment, or any other amino acid sequence of the same length. And what is important, we cannot do this without modeling. What do I mean? If you, for instance, take a coin and you want to have the probability of head and tail, you just repeat, draw the coin many times, and you count the head and you count the tail, and then you know something about the probability. And if you do it frequently, you will directly by counting get the probability of head and tail. Here, each sequence we have only once, and most possible sequences have never been seen. So we have a very small sample of something which is full probability distribution. So we have somehow decided on models, and one way which is used quite a lot in the field is via so-called maximum entropy modeling. And the idea is that instead of using the data itself, we decide on a certain number of observables which we make on these data. So these are functions, real valued functions basically of sequences. And then we say that our model should reproduce the average observable. So we have here the average of the observable over my data set, and I would like to have the model such that it reconstructs, that it gives again the same, so the ensemble average in the model is equal to the empirical average over the data. And via maximum entropy, this tells us that the Hamiltonian of this model should be a linear combination of the observables with corresponding Lagrange multipliers. Obviously, the precise shape of this model depends crucially on what we decide are the observables. So we need some intuition to find good observables. If we do a bad job in choosing observables, everything will be completely useless. In the case of proteins, one kind of observable has been used a lot over the last 20-30 years, which is so-called conservation, and which is what I already mentioned. I told you that there are some columns which are use only one letter, one amino acid, or almost one, so in 90% it's one, and in 10% it's another one, and others change a lot. And this can be captured in one observable, which is the fraction of sequences having amino acid A in column I. So it's something like a local magnetization, basically. And if you want to have a model which actually reproduces these characteristics, you find something which looks like a model of independent spin in a local external field. So in computational biology, these models are called so-called profile models, and they talk about independent positions. These models are a very nice example of what actually is this wrong models, which are extremely useful. Because they are actually basically the most successful modeling approach in bioinformatics so far, because so these profile models frequently are represented in a way like here with what is called a logo, and you find and basically the height of so each position here corresponds to one position, to one column in the alignment, and the height corresponds to the conservation, or more precisely to the information contained in the position. So if I tell you position 11, you know it's probably histidine. That's why there's a big H here. And so these models identify these conserved positions, and these conserved positions are frequently very important, either functionally. So for instance, here this histidine is a functionally important active site. If you change it by something else, the protein does not work anymore. Or structurally important. These models are actually used to identify homologous sequences. So out of all these 100 million sequences I was talking about, those which are put into one protein family is done with this kind of models, and it's used to align sequences. So the sequence alignment, the multiple sequence alignment I showed you in the first place, is actually constructed using these kind of models.
different proteins are not the same.
Yes.
Okay. So why the position? So, I mean, it could be that you add something in the beginning or the end of the sequence.
Yeah.
So the position is not invariant.
So one important point in aligning sequences is that actually the length is not invariant, and sometimes we have a loop in a protein where you can add or subtract 1 or 2 amino acids. And this is actually taken into account in what I have here with these little minus signs, which are alignment gaps. And the alignment is actually done in a way to have a maximum of conservation. So basically you have some points like this histidine here which serve as anchor points, and others, and you try to make them as similar as possible in the middle, allowing for either skipping some positions which are called insertions, or by introducing this minus sign in case of what is called a deletion. So the multiple sequence alignments are constructed in a way to have this variable sequence dealt with. So the letters in our sequence alignment are actually the 20 amino acids and this minus sign for the insertion and deletions. And you see there can be quite consistent, quite large parts which are actually gap. Okay. So I told you that these models are extremely successful and extremely useful, but they are wrong. They are wrong in the sense that they miss important information which is contained in the data. And one, for instance, they are, since they talk about independent positions, they are unable to capture any relation between positions. But for instance, if you talk about in a fold about 2 residues which come into contact in a fold, that's somehow a relation between positions. Or if you talk about protein-protein interactions, so 2 protein coming into contacts, they have to be compatible to actually come into contact, and all this cannot be captured by these profile models. So another kind of information which we try to take into account is what is called co-evolution, and it's a very simple concept in principle. Imagine that you have 2 residues which are here in contact in my cartoon protein, and to be in contact they have to have somehow compatible physical-chemical properties. Now if I mutate one of them, it's pretty likely that I destroy this compatibility. But maybe by making a second mutation in the second one, I can compensate for what I did before. So I can re-establish this compatibility, and the protein is again okay. So if you have a situation like this, you could have something like in this simplified, completely fake sequence alignment here, that you have, for instance, RD together, you have EK together, but you don't have RK and ED. In terms of statistics, you have to do 2-point statistics. So you ask how many sequences actually contain RD together. So this would be 50% here, and RK would be 0%, and so on. And if you want to establish also, if you want to reproduce this observable also in your models, then necessarily you have to introduce couplings between residues. So you have to have a model which basically looks like a generalized Potts model in statistical physics. So you have these couplings, which we call direct couplings, and the inverse problem we named direct coupling analysis some years ago. Now, these numbers are directly, we can calculate them directly for having the multiple sequence alignment. It's basically counting with some little statistical tricks. But calculating these ones here, so we have to fit these couplings and these fields such that we reproduce actually these statistics. And that's a complicated problem. It's a complicated problem because we need to be able to calculate observable thermodynamic averages over basically arbitrary Potts models. And we don't know, we know that we cannot do this, and here statistical physics has helped a lot in establishing approximations based on mean field approximation. We have worked with message passing, which in this case didn't work too well, but approximations to actually make this relation between these couplings and the observables, and to be able to fit these two data. So I don't want to go into details there, because I want to stay more at the level if these models are useful and what can they be used for. And actually, the model which I have shown to you in a more formal language would be a graphical statistical model. And as graphical statistical model, it has at least two aspects. One, it's defined on something like a graph or a network. And is this graph and network, is the topology of this this object somehow informative? Does it contain some interesting biological information? And then in a more fine scale way, we can ask, now we give a probability or some kind of a statistical energy to a sequence. Does this number mean anything? So what's actually the biological information which is in these models? And the first thing I want to answer very fastly is actually this, the fact that these strong couplings, so the topology of the network basically predicts the structural contacts in the folded protein. What I show you here is two plots. So it's one specific protein. The trypsin inhibitor is a small protein, but apart from being small, it's a relatively typical protein. And I just, on top of the known structure, in this case, I plotted what comes out of the sequence analysis. In the first place, I just looked to correlations. So basically, I just, without doing any inference, I just looked if this Fij is different from Fi times Fj. And I plotted the first 30 most correlated pairs, and you see that these most correlated pairs, some are very distant. Actually, the red ones are really in contact, and the green ones are distant. So it's a wild mess. Whereas if you go to this inverse problem, if I take the first 30 couplings, they are all in contact. And this was quite a surprise for the computational biology community, because it's something which works quite consistently for many, many protein families. And I just, and it's widely used now for predicting three-dimensional protein structure from sequence. And I just want to mention this last paper from David Baker's group, which came out last year. And what they have done is basically, I told you about these 16,000 protein families which are known. About 4,000 of them have no known protein structure. Not even a single protein out of the alignment has a known structure. So these are kind of the targets of some studies like this. And what the Baker group has done, they have assembled as much data as they could. All sequencing projects around this planet, they have tried to get data. So they have compiled big alignments, and then they have just used this methods to do structure prediction. And this is a single paper. Out of these 4,000 families which have no known structure, they were able to predict structure for almost 500. So that's almost 12% of those which were unknown were predicted in this single paper. So typically one prediction, one paper. So they could have made 500 papers, but they did one single. The quality of these predictions is, can be discussed, and so in some cases, but still it's quite important. You can use it, and I want to only mention this shortly, also for protein-protein interaction. And in case of protein-protein interaction, what you have to do is you have to have something like a joint multiple sequence alignment, where each line contains two proteins which interact. And then you can do exactly the same statistical mechanics formalism. There's no difference between the alignments at the end, at the formal way. And you can learn quite a lot about protein-protein interaction networks and about contacts in between protein and the assembly of protein complex structures. Okay, so hopefully you believe now that this network of the graphical model is informative. But as I told you, there should be more quantitative information because we can actually calculate the probability for each single sequence. So what can we do with this probability? And in the last 5-7 years or so, a new experimental technique came up and became quite popular, which is so-called deep mutational scanning of proteins. Basically, the idea is to take a protein, to make all single mutants, so in each position you have 19 mutations, or to make a large set of mutant proteins which can contain double mutants, triple mutants, and so on, and to try to measure the effect of the mutations on the proteins. And we started a collaboration with Olivier Tenaillon, who is in Paris also, and who made one of the first big data sets there in a protein which is so-called TEM1, which is a protein which provides antibiotic resistance to E. coli against amoxicillin and ampicillin. And it's actually an extremely successful protein. It's not in the genome of E. coli. It's on a so-called plasmid, which is a small circular DNA, and it can be either in an E. coli or not. And the difference is basically if it's resistant or not. And amoxicillin was introduced in 1960, and in 1964 or so, the TEM1 was first reported in a few strains of E. coli. And now about estimates are between 1/3 and 1/2 of all E. coli strains, so including all those in your gut, for instance, have now this protein, this TEM1 protein. So it's an extremely successful protein. So what they have done, they have introduced random mutations into these proteins, and when they introduce random mutations, obviously there are a lot of proteins which don't take any mutation, so it's basically almost a Poisson distribution of mutations, and there were about 1,000 single amino acid changes, and for each one they have measured the resistance to amoxicillin, which is basically measured by something which is called minimum inhibitory concentration. It's basically you try to grow the E. coli with this mutant protein in different concentrations, and the minimum inhibitory concentration is the smallest concentration where it stops to grow. So at lower concentration it grows, at higher concentration it does not grow. I told you that there were quite some proteins without mutations, and these are plotted here as the white bars, and you see they are quite peaked around a minimum inhibitory concentration of 500 milligram per liter amoxicillin, with some error bars around. But if you, the other plot here is the single mutant effect, and you see they have a much larger spectrum. Basically, a lot of mutations are quite bad for them. So they have a much lower minimum inhibitory concentration. They basically lose resistance, or a big part of the resistance, whereas we don't find anything which is really better. And actually single mutants don't help these proteins to get better for some more complicated reasons. So now we wanted to see if this has any relation with our models. And the idea was, okay, we do exactly what I told you before. We take this protein, TEM1, we can construct its protein family. It's actually one of the 16,000 family. It's a number 13,354. Okay, that's a protein family. We construct this sequence alignment, which when we did this work had about 2,500 proteins, which are very, very divergent between each other. They are at about, on average, 20% sequence identity, 80% sequence divergence. We constructed this model, as I told you, and now we can score on our computer mutations by just calculating the probability of the mutant with respect to the probability of the wild type. So taking the logarithm, we have something which is a positive score if the probability goes up, so maybe it's a beneficial mutation, or negative if the probability goes down, so it's probably a deleterious mutation. So this would be our expectation. Still, I remind you that we have done this inference on these very, very diverged proteins. So somehow maybe we capture something which are evolutionary constraint with are valid for an entire family, for very distinct proteins. What was measured in the experiment was one specific protein and single mutations. We have no sampling of anything which is so close. So do these have to do something with each other? And the answer, which we didn't have before we tried it out, is actually that there is a lot of information. And in particular, if you compare, so what I plot here is an R-squared value. So it's a square of a linear correlation, of a correlation between what our model predicts and what the experiment does. So it's what is typically called the variance explained by our model. And you see there's something like 40% by the profile model. This is something which is quite understandable. If I mutate a very common amino acid in one position into something which I've never seen in any protein, probably it's bad. So it's expected that you have something. But if you take this model which has the couplings, you have a much higher value here. And other modeling approaches which are based on structure, for instance, like this one here, are much worse. The ones which are somehow comparable are both based on profile models. So SIFT is basically equivalent to the profile model. It's profile constructed a bit differently mathematically, but the same idea of independent sites. And PolyFan2 is a supervised approach which takes basically profile models together with structure and tries to do some supervised learning on top of them. Okay, so there is also some information in this probability vectors actually, and so we see that these couplings actually add something very important. So it's necessary to put them into good models, but there was also a question, do we need to go on? Do we need to put something like 3-point couplings and so on, or are these 2-point couplings something like sufficient? And there was a very nice paper by Rama Ranganathan's group in 2005 where they did some very cool experiments, pretty small scale. So each of these plots here is 40 proteins about, and on a very small proteins of a bit more than 30 amino acids, which was possible at this point. And what they did is basically they took first natural proteins. So they took an alignment of about 40 natural proteins and they measured if they fold or not in some way. And the red part are those which fold, and the others have problems. There may be problems. They are not folding. They are not even expressed in the expression system they use. They are clumping together. They are agglomerating, so they are not solvable in water. But you have this 2/3 which work. Then they completely scrambled the alignment. Random, nothing works. Then they tried to do some scrambling, which is a bit more intelligent. They scrambled columns of the alignment. Columns of the alignment mean that they completely maintain the profile. They maintain the conservation statistics. Nothing works. And then they did some kind of Monte Carlo procedure, which actually scrambled still columns, but in a way to re-establish the covariance matrix between different columns. So something which resembles pretty much to what we do. And here you see that a quite large fraction, 28% of them folded. So this is quite remarkable. And here you see one of these proteins resolved by NMR spectroscopy, the structure, and here an overlay over a number of natural protein structures, and you see it's the same. So it actually works. What this paper was not able to say is why these 28% work and the others not. We could just construct a new artificial sequence alignment and then do the test. But we have a model. We have a model which gives an energy to, or the statistical energy to each sequence. So we can look to this plot here. We have plotted the statistical energy of the model. So low energies obviously are high probability, and high energy is low probability. And all these lines here are in a sequence of the experiment, and they are plotted in red if they are folding, and in gray if they are not folding. And what you see is that actually this randomized and the first two kinds of randomization, the one respecting or non-respecting the conservation, they have substantially higher energies than the natural ones, and that's why they don't fold. But now the astonishing part is if you look to those which they have randomized, trying to get back the covariances, you see that the red lines are almost all down here at low energies, at energies which coincide somehow with a range of energies found in the natural ones. Whereas those which have higher energies, they typically don't fold. So this looks this made us a bit optimistic to start another project, which is on statistical protein design. From a statistical physics point of view, it's almost trivial. I just take my model, which I have inferred, and I do some Monte Carlo sampling. And like this, I have designed new protein sequences. So it's very trivial. To be able to work, it should be something which is which we could call a generative model. Generative model basically means that if I give you a mixture of these sequences and these sequences, you cannot tell them apart. So I could give them to Guilhem, to Florent, and so on, and Andrea, and many others. You should not be able to tell them apart if you have a good generative model. So statistically, they look the same. This does contain, obviously, in the first place, very precise fitting. I told you that we fit 1 and 2 point frequencies of the alignment, and doing this with much care in the models, and we need to do some quite stupid methods based on Boltzmann machine learning, where we have to sample the distribution, adapt parameters, sample the distribution, and so on. It's quite expensive in terms of calculation, but then we can fit very well the 1 point which are in this kind of reddish color here, and the 2 point correlations, which are blue. But okay, that's not astonishing because we fit them. We just did well our work. What is more astonishing is actually that we reproduce statistical properties which we did not fit, which we never gave to our algorithm. The first one here are 3 point correlations. So there are 3 point correlations in natural sequences. And if you take a profile model, obviously all spins are independent. You don't have any 3 point correlations. And actually you find a flat line here up to some finite size fluctuations of the finite sample fluctuations. Whereas what we get for our pairwise model is actually very correlated with the 3 point correlations. Another observation is, for instance, that natural sequences tend to be clustered here, represented by the two first principal components of the sequences. They are somehow clustered in sequence space. A profile model does not see this clustering, whereas our model reproduces quite well this clustering. This has never been given to the inference algorithm. So it seems to be something significant. It's very easy to give you a data set where you completely fail with pairwise models. The simplest thing is for 3 variables, just a parity check. You have no magnetization, no 2 point correlations, and so you would completely fail. So, but still, these models are not as good as one might think after this. For instance, they have, if you sample, they don't fall into the good energy range. Here I have, for this one protein family, I have made a histogram of all energies in our model, and this is a sample. The green one here is a sample from our pairwise model. And you see they overlap, but they are shifted somehow to higher energies, to smaller probabilities. They are better than if we, for instance, make some random sequences or some profile model sequences. They are worse, but still we are significantly higher. And the reason for this is very easy. The reason is that we need to use regularization, and actually regularization makes that the statistics of the data is not exactly the same thing as the statistics of the model, but they're shifted by some regularization parameter times the parameters, which leads to actually a shifted energy. And we have to take this into account in our design strategy. So what we have done, I can be very fast on this experimental part. These are experiments done by Bill Russ and Rama Ranganathan in Texas. And it's done on a protein which is a course-mate mutase, which is about the length 100, slightly left 96 or so. We have used 1,200 sequences, and it is an enzyme, a biosynthetic enzyme, doing amino acids. So you need it, E. coli needs it, for instance, if these amino acids are not provided in the medium. So we have done what I told you, and then experiments are done, and the experiments are growth experiments where you have E. coli bacteria, which are genetically identical up to the single sequence, where the natural sequence is replaced by my sample, my Monte Carlo sample. And then I let them grow, and I sequence before and after, and I count how frequently my sequence I find them at time t compared to time 0, and compare this to the wild type E. coli sequence at time t and time 0. So this is a relative enrichment. If it's around 0, it is as good as in E. coli. If it's strongly negative, still these models are wrong. I don't have the time for doing the conclusions. There is a lot of things which should be done. I would like to thank a lot of people who have contributed in one way or the other to this one. And I would also would come back to what I said in the very beginning. So even if I always state at Giorgio distance 2, correlation can be also arise from indirect, without direct interaction. And I have just listed, I don't know if it's exhaustive, the list of the common co-authors. And this has triggered some very big correlation. In this case, it's clearly directed, and that's why thank you and happy birthday.
Thank you, Martin. Since lunch is waiting, one or two questions? Go ahead.
So when you do the sampling of your sequences, you have shown that they are clustered. So it looks like that it could be a glassy system. So it's actually hard to sample it, or is it just because they are so small that you don't have sampling or equilibration problems?
So it is ergodic, more or less. Even if you see in this plot here, if you sample at lower temperature, it gets fragmented where you sample. So the colored ones are the sampled ones. And actually we need the regularization to make it ergodic. If we wouldn't do regularization, then then it becomes non-ergodic and we cannot do the inference. So we have to keep it kind of still doable.
Yes, there exists a method called multi-scale relevance, which should provide the most informative sufficient statistics that the maximum entropy method should reproduce by construction and given a database. And I was wondering whether this method has been used in this context or it could be used, and whether in this case it would say that the most relevant sufficient statistics is indeed the average, I mean the local magnetization and the pairwise correlations.
I'm pretty sure that, so we decided on these pairwise statistics just in a very heuristic way. because single was done. We had enough sequences to measure pairwise, so let's implement them. Actually, this causes a lot of problems because we have many, many parameters, and we need this regularization and so on. I don't really know of anything which really can tell you which are the relevant, which is the sufficient statistics, because we assume a sufficient statistics. It seems to be that the pairwise do a quite good job in the sense that you reproduce statistics which you didn't learn, but you put too much. You put far too much. And there are ways to do some sparsification or dimensional reduction and so on self-consistently, which actually show you that you can do an as good job with much less parameters. But so far, I don't know of examples where you can actually do a better job, because having less parameters, you could regularize less and maybe do better models.
Perhaps the multi-scale relevance method is suitable for this kind of problem.
Sorry?
The so-called multi-scale relevance method, it should give you on information theoretical grounds which is the sufficient statistics. So perhaps it could be relevant to this problem. I don't know.
You should try.
Okay, then we thank everybody and we go for lunch.